Hello students, welcome to another video of educators on absorption of fat. Yesterday I have put forward a video on digestion of fat. Now today we will know about the process of absorption of fat in our body. It is considerably a bit complicated phenomenon. Okay, the absorption of fat is a bit complicated phenomenon, and this takes place in the cells of the intestine with the help of several enzymes and digestive juices. So the most common hypothesis, there are many hypotheses in the absorption of fat, but the most common are given by the Fluger, Verger and the McDowell, who gave the lipolytic theory. And Fraser is the one who proposed that the absorption of fat is more or less like a the partition hypothesis. Okay, so how absorption of fat takes place after the fat molecules are broken down into fatty acid and glycerol. We'll be learning that in detail in today's video. So let's move on to our video. Welcome to Educators once again. Don't forget to like, share and subscribe the channel. Over here we can see that the digestion of lipids. So we in the last video we have um, stopped till this portion where the lipase enzyme has broken down the smaller droplets into glycerol and fatty acid. Now what is the fate of this glycerol and fatty acid after it is broken down from the fat molecules? So digestion of lipids include uh, the bile salts which break the large masses of the fat into smaller droplets, and finally the lipase enzyme break them down into glycerol and fatty acid. Now they can be formed in any of the emulsified form, or they are absorbed as a fatty acid and complex bile salt and fatty acid complex. So let us learn before we move into the detail of the fat absorption, let us first know what is this micelle and what is this chylomicron. So these two are very important in fat absorption. So what is this micelle and what is this chylomicron is a very important part. So micelle is nothing but produced with three major components. One is the bile salt, the monoglyceride and the fatty acid, which are again combined with the phospholipids to combined with the phospholipid molecules. How much uh, bile salt, the amount of bile salt necessary for the formation of micelle is known as critical micellar. So the amount of bile salt which is required to form a micelle is known as the critical micellar. Okay, critical micellar. Now these are some compounds with 4 to 7 nanometer in they are small lipid droplets, and they have, as you can see over here, they have a hydrophobic core area and hydrophilic shell area. Along with that they have lecithin, cholesterol, lipid soluble and also the bile salt, which help the fat molecules to pass from one to the other part of the absorptive stages. Okay, and now coming to this chylomicron. What is this chylomicron? As you can see in this image, chylomicron is also a compound made up with cholesterol, phospholipid, protein, which is actually covering the fat droplet or the triglyceride. So it is basically of 1 micron in size, in diameter rather, and are extracellular fat droplets. They don't contain only triglyceride but also composed of protein, as I have shown in the picture. some free cholesterol, and saturated triglycerides, and monolayers of phospholipids. So everything is there in the chylomicron compound. Now, what is this chylomicron? Where the chylomicron helps, we will come to know, but then also I'll just tell you that this chylomicron are released from the epithelial cells, and they pass into the lacteals and the lymphatic capillaries in the intestinal villi. So that is what where a chylomicron is required or necessary. Okay, so micelle and chylomicron, what are these structures we know. Now we will come to the absorption of fatty acid and glycerol. So once the digestion of fat takes place and we are getting glycerol and fatty acid, what happens to this glycerol? What is the fate of this glycerol? Some glycerol are partly oxidized to carbon dioxide. Some are used in the synthesis of glycogen, and some are resynthesized to triglyceride. Now, when I'm talking about resynthesis to triglyceride, then you must be thinking that why then it is being broken down into glycerol. Again, it is being resynthesized to triglyceride or any other part of the fat digestion, because they are covered by the micelle. The fat molecules are covered by the micelle, and they need to be uh, just cross the epithelial cell, and once they are absorbed in the other part of the epithelial cell, micelle can't pass. Okay, so they just form the micelle to cross the barrier for the diffusion, and then they are again resynthesized or broken into fatty acid, glycerol, and few amounts of monoglyceride. So this is where the resynthesis to triglyceride is very, very important. So this is the final slide of the absorption of fat. So we know what is the fate of glycerol. We know what is a micelle, what is critical micellar, what is a chylomicron. So we know these small part. Now we will assemble everything together for the fat absorption. So what happens? We know that we are getting fatty acid and glycerol after the digestion of fat, which are emulsified by bile salts, and micelle is produce. Now the thing in micelle is that it continuously being formed and again denatured, again being formed and again breakdown takes place. Okay, so this is you can say a type of micelle. Why? Because micelle is only required to make them absorb through the villi of the epithelial cell. Just to do that, micelle is required. So in the previous slide only we have seen that how in what a micelle is formed of. Now this micelle, they take the fatty acid, glycerol, glyceride, monoglyceride together. They cross the barrier. Micelle, the bile salt is again reabsorbed, and it is again broken down. Broken down to form what? The fatty acid separates, the glycerol separate, and the monoglyceride separate. Now, when the fatty acid and monoglyceride separates, they form the triglyceride. This triglyceride, they use the phospholipid, they use the protein from the Golgi bodies, and they form the chylomicrons. Now these chylomicrons enters the lacteal and are transported away from the intestine. So this is how the fat absorption takes place. In the right hand side photograph or the right hand side picture, the same thing is given, but it is more point wise. So first step is what? Bile salt, fatty acids, and glycerol, they are forming this micelle. This micelle is what is doing? The bile salt along with fatty acid, they are simply moving into the epithelial cell. See, once the epithelial cell is there, the bile salt is reabsorbed. Over here the sign is not given, but actually the bile salt is reabsorbed, and the micelle holding the fatty acid, monoglyceride, and the glycerol, they enters as fatty acid, monoglyceride and glycerol and forms the triglyceride with the help of simple diffusion. Again, as they move forward, they are protein coated from the Golgi bodies, as I've just said, and they form the chylomicron. Now, there are certain patterns like fatty acid of less than 12 content, fatty acid less than 12 carbon content, they move directly to the blood. But fatty acid more than 12 carbon content can't move to blood directly. Now why? Because the slit of the chylomicrons are the slit pores, they have large slit pores, okay, which help them in between the epithelial cells and the chylomicron. So that helps the chylomicron to pass through the lymphatic system, through the lymphatic system, from the lymph to different parts of the body, and then from there to it comes to the systematic blood, and to the different systemic blood, and to the different parts or organs of the body. So this is how the whole process of fat absorption takes place. So chylomicrons, they are released from the epithelial cell and they pass into the lacteals along with the lymphatic capillaries in the intestinal villi, rather into the blood capillary. Why? Because they don't, in connection of the lacteals, they don't have basement and have very large slit pores. As I've told, they have very large slit pores which help the fat molecules, rather the chylomicron, to pass into the uh, lymph through the lacteals, lymphatic system through the lacteals, rather than the blood vessels. From the lymph, I'm just repeating it again, from the lymphatic system, they again move back to the systemic circulation and to the parts where the body requires, or the organs requires, fat. It's a bit complicated, but I hope you have got this absorption of fat. I have tried to make it simple. So that's all for today. Thank you for joining, educators. Stay tuned for more videos.